Great. Are you ready to get this show going? Hi, everybody. Welcome back. We're going to start our next presentation, so if you could please take your seat, that would be great. It is my pleasure to introduce Dr. Ignacio Garrido-Laguna with the Phase 1 Program Director at the Huntsman Cancer Institute. He will be speaking to us today about the trends in Phase 1 clinical research design.
Good morning, everyone. I hope everyone is having a great conference. I want to start by thanking the organizing committee and our chair today for the introduction. I was asked to say a few words about myself, so I'll start by saying that by now, most of you have realized that I'm not from Utah, right? And that I didn't even grow up speaking English, right? And if you didn't yet, thanks for your kindness, but check your pulse, because I'm gonna try to do my best here, but I always, after so many years in US, I always say it's not the same when you didn't were raised with the language, right? So those of you who want to know how good is my Spanish, because that's the one thing Spanish do very well, we speak very good Spanish, just come to the Spanish track sessions later today. So I'm a Phase 1 investigator at Huntsman. I see some of my colleagues here. See Rachel Kingsford sitting right there. And I run a large group where we try to provide new treatment options for patients who essentially have exhausted therapeutic options for their disease. We have a large group, work very close to clinical research coordinators, research nurses, RDCs, specimen processors. And as a PI, I fully appreciate the amount of work that goes to you, right? And that the success of the clinical trials relies a lot on the quality of your work. So I want to say thank you to all of you for everything you do every day for our research programs, but most importantly for our patients. These are my disclosures. None of them are really relevant to my presentation today. And this is going to be the agenda. I'm going to walk you, try to give you a historical perspective of Phase 1 clinical trials, putting some emphasis on the low response rate in traditionally Phase 1 trials without a biomarker selection, but also on the high attrition rate that we see in oncology. I'm going to spend a few minutes talking about the different paradigms that we have been using with cytotoxics, with traditional chemotherapy for many years, impacting significantly the Phase 1 trial design, and that unfortunately we have continued to apply to the development of novel therapies like molecularly targeted agents. So I think it's important we understand the differences between this class of therapies, because that provides the rationale to implement, to develop Project Optimus. We'll spend some time also discussing how Project Optimus has impacted the design of clinical trials. And finally, a slide to summarize some of these thoughts and give you some thoughts for the future. This is a study that came to my attention like 16, 17 years ago when I was a fellow at the Phase 1 program at MD Anderson. Essentially, they look at Phase 1 trials that had been conducted through the decade between 1990 and 2002, and they look at the response rate when new drugs were given as single agents. What you can see on this slide is that responses were nearly negligible, 4%. These studies were mostly designed to understand safety and toxicity with little hope that we will see therapeutic activity. Obviously, things have changed a lot in the last 25 years, and you know that many of the drugs, or several of the drugs we have in the clinic have been approved based on activity shown in a Phase 1 clinical trial. More recently, this is a meta-analysis conducted enrolling around 6,000 patients conducted between 2015 and 2018 including close to 160 trials. And what you can see in this meta-analysis is that when we have a biomarker, when we can identify the patient population more likely to benefit, the response rate increased by a factor of 3 compared to the prior study. You may say this is not great, and I agree with you. This is not great. And not surprisingly, attrition rate in oncology is the highest among the different medical specialties. Only 1 out of 200 drugs that make it to preclinical stages of development will eventually become approved by the FDA. Unfortunately, lack of activity is often not detected at early phases of development. Many of these compounds get to a phase 2, phase 3, and that's the point when the drug is deemed to be non-active. By then, we have already treated hundreds or thousands of patients with a drug that has not made significant impact in their life expectancy. So what are the most common contributors to the high attrition rate that we see in oncology? Suboptimal preclinical models. Think about these cell lines, some of them obtained from patients back in the 50s or 60s, and then passed through multiple passages in plastic over the years. Those cell lines do not really recapitulate the disease biology any longer. Think about the xenografts, the mice models derived from those cell lines. They really don't recapitulate the disease biology. A lack of validated assays to measure on-target activity and to understand what, if anything, the investigational drug is doing to the sites of disease. Suboptimal understanding of the target or the patient population more likely to benefit from that investigational agent. For many of these investigational drugs, we lack a predictive biomarker. We don't have the strong science or validated biomarkers behind. Resistance, and particularly with some of the drugs that you have seen recently raising a lot of excitement in the clinic, like the new RAS inhibitors, adaptive resistance, it's a significant issue. It means that when we are inhibiting the pathway, the RAS pathway, there's alternative signals that are triggered that activate a receptor at the level of the cell membrane that reactivates the pathway. That's why those of you who have worked with G12C inhibitors in the clinic have seen how we have added antibodies targeting EGFR to try to overcome resistance. But resistance, multiple types of resistance, genetic, non-genetic, continues to be an issue. And finally, toxicities. Often, we don't have the right dose or the right schedule, or we have not been able to refine the dose and the schedule for our patients. I think it is a significant failure when we have an active drug and we are not able to get a schedule and dose that allows our patient to continue treatment to be able to see the full potential of that drug. This slide is just to show you how in the last years we have seen an increase in post-marketing requirement studies that are designed to evaluate a lower dose of the drug, lower than the one that had been approved by the FDA. This sends a strong message. We're not doing great at finding the right dose for our patients. And that's the result of using all paradigms from the cytotoxic era that have been applied to the development of molecularly targeted agents. Here, I just give you a few examples of drugs that were FDA approved at a higher dose that eventually we identified was just too high for our patients. Ponatinib, small molecule approved for patients with CML, chronic myeloid leukemia, associated to thrombotic events at a higher dose of 45 milligrams daily. Subsequent studies showed that the drug dose could be reduced to 15 milligrams once those patients have attained a molecular response to therapy. And why is all this? For many years, we have been using paradigms from traditional chemotherapy, from cytotoxics. And that's what I'm trying to show on this slide. And that has permeated to the development of novel agents. On the right side, I wish I could use pointers, but we'll do it this way. If you look at the plot, on the right side, the dashed line shows you the dose toxicity curve. And what you can see there is that there's a step relation, right? Step curve. The higher the dose, the better the activity. And that curve goes up very quickly. With small changes in dose, you have substantial increases in activity. But if you look at the solid line on the right side, we see also significant increases on toxicity that follow the increase in the dose, that are clearly dose-related. So with cytotoxics, patients experience toxicities rapidly, within a few weeks, and they might be willing to tolerate those toxicities because we tend to use shorter treatment courses. We treat these patients for just a few months. We're not thinking giving them chemotherapy for years, like we do often with molecularly targeted agents. So patients are willing to deal with some of these toxicities. One of the problems with these models, like the 3+3 design that have been traditionally used as a dose escalation design in phase 1 trials, is that we use short DLT windows. DLT windows stands for dose limiting toxicity window. That period of 4 weeks where you see on your protocol if patient has a grade 3, grade 4 toxicity that meets protocol criteria, we will consider that a DLT, and it will tell us that dose might not be well tolerated. Unfortunately, that same paradigm doesn't apply to molecularly targeted agents because some of the toxicities we see with small molecules that impact significantly quality of life do not happen during the first 4 weeks of treatment. So the higher the dose, the better, right? Yes, of course. And we have seen that also with cytotoxics. That's what I'm trying to show on the right side of the plot. This is a study for patients with resected breast cancer that received adjuvant chemotherapy. Solid line on top, it's higher doses of chemotherapy. Dashed line below, lower doses. Patients treated with chemotherapy at higher doses had improved disease-free survival, indicating or suggesting that at least for chemotherapy, the higher the better. Now, what is the problem? Most of the drugs we have these days in clinical trials are not cytotoxics. You can see blue line and orange line on top, and then cytotoxics is that blue line in the middle, so gray and orange on top. Those are molecularly targeted agents or immunotherapy. And if you work in clinical research units, you may have noticed most of what we get these days are immunotherapies, and of course we still get quite a bit of molecularly targeted agents. So what is that I call the modern sophistry? The modern sophistry is the application of paradigms that were developed through the cytotoxic era to continue to apply them in the molecular targeted therapy era. So Protagoras, Thrasymachus, and Gorgias were sophists. And what sophists will do is just use their rhetorics to persuade crowds rather than to pursue the truth. And they will use premises that may look like true, but they were not accurate and will lead to the wrong conclusions. So premise number 1, anti-cancer drugs work best at maximum tolerated dose. Yes, that was true maybe 20 years ago, not true anymore in the molecular targeted therapy era. Premise number 2, molecular targeted agents are anti-cancer drugs. That's true. And now you replace anti-cancer drugs by molecularly targeted agents, and you get to the fallacy. Molecular targeted agents work best at MTD. That's false. We know that's not true. And even so, we have been developing these drugs to try to get to the MTD. And I recognize that there's some advantages of understanding what the MTD is, and I will discuss later. But we have failed to identify a range of doses that might be efficacious and may lead to better treatment compliance for our patients. So what are the paradigms from chemotherapy that don't apply to molecularly targeted agents? PK variability. With chemotherapies, most of these drugs are delivered IV. So there's really not absorption that limits significantly the variability. With molecularly targeted agents, depending on the pH of the stomach for some drugs, or depending on other factors, whether you take the drug with food or fasting, we may have substantial variability in drug exposure, even at the same dose level. The therapeutic range. With cytotoxics, the therapeutic range is narrow. Small changes in doses are going to lead to substantial increases in toxicities. With molecularly targeted agents, the dose-response curve, it's flatter, it's not a step. That gives us an opportunity to select a range of doses that really have very similar efficacy, but may have significant differences in toxicity. The dose paradigm related to this I just mentioned. With cytotoxics, we're looking to those close to the maximum tolerated dose because we're looking for highest activity, and there's huge variations in response due to small difference in doses. We don't see that with molecularly targeted agents. The DLT definitions on the protocols and the window for DLT. With cytotoxics, we only care about severe acute toxicities, grade 3-4 events. With small molecules, we care about low-grade chronic toxicities, that grade 1 skin rash or diarrhea that patient can tolerate well for 4 weeks, but it become an issue significantly impacting quality of life when persist for several weeks or months. So it is about finding the right balance between efficacy and toxicity. And the right balance might not be where this arrow is sitting. It's not the dose that leads to the highest efficacy when that also leads to substantial toxicities. We may need to sacrifice a little bit on efficacy to gain on substantial decreases in toxicity. I mentioned earlier about how drug exposure with oral drugs might be affected for multiple reasons. This is just to emphasize that. Pharmaceutical formulations, I think, or even the capsule burden, right? We see on some of our phase 1 trials. We have a drug that we have to dose, giving the patient 8 pills in the morning and 8 pills at night. That's not a successful path for good treatment compliance. Pharmacogenomics. Patients may have genetic polymorphisms in CYP3A4 or other enzymes that alter the drug metabolism. Drug-to-drug interactions. Think about, for instance, small molecules we have in the clinic, like tyrosine kinase inhibitors. These drugs are weekly basis. And if we give a PPI that alters the pH of the stomach, and the stomach is less acid, the drug is going to be in the non-ionized form, and we're going to have decreased or compromised absorption. Same with food. Dosing with food may increase the drug concentrations of a drug. We know that with grapefruit, for instance. So you have read probably about strategies to give lower doses of a drug together with food and having similar PK profile to that of higher doses of the drug given fasting. And then, of course, differences in absorption, distribution, metabolism, or excretion. So what studies aiming at maximum tolerated dose are missing is that there might be drug activity across multiple dose levels and not just at the maximum tolerated dose. That range of doses is what we need to understand from phase 1 trials. And on the novel designs, that's what we're looking for, trying to identify a range of doses so that with additional information about the drug exposure, the response rate across those doses, and of course the toxicity, we can make a decision about what is the optimal dose of the drug to move forward on clinical trials. I love this picture, and I love it because it absolutely highlights what Optimus is all about. So this is a study with infigratinib. This was a FGFR2 inhibitor that was developed and approved by the FDA for the treatment of patients with intrahepatic cholangiocarcinoma with FGFR2 fusions. On the x-axis, you have treatment duration in days. On the y-axis, you have each of those lines, it's a patient. Each line has been color-coded according to the dose that that patient was studied on. Blue color, 125 milligram, yellow color, 75, and some dose in between. The red bar I plotted to show you a trend. Left of the red bar, you see mostly blue color. Those patients were able to tolerate the full dose of infigratinib, the FDA-approved drug, for 48 days. After 48 days, nearly everyone needed dose reductions, dose modifications. Despite it was an active drug, patients were not able to tolerate the low-grade chronic toxicities, the stomatitis, the paronychias, the eye toxicity, etc. So the FDA required a post-marketing requirement study, and eventually the sponsor withdrew the label, understanding how competitive this market was and how difficult it was to conduct a post-marketing study once a drug is already approved by the FDA. Why would a patient take the trouble to enroll on such clinical trial if the drug is already approved? Why would they travel? Why a research site allocate resources to do this with all the complexity of this, participating in clinical trials? So the promise of precision medicine is not only to give the right drug to the right patient at the right time, but also to be able to deliver the right dose that is going to maximize therapeutic activity and minimize toxicities. Optimus is not new. 30 years ago, the International Conference for Harmonization issued this document. And I'm going to read this. Dose response assessment should be an integral part of drug development. And we have ignored, or ignore it's perhaps a little bit hard, but we have not been able to deliver on this over the years. So most recently, as you know, the FDA started Project Optimus, and the last update was just 4 or 5 weeks ago, which is what I'm showing there on the right side, stating sponsors should carefully evaluate exposure response, efficacy, and safety data from early trials to inform dose selection. So it's not about toxicity alone anymore. It's about the totality of the data. I'm not going to read this slide to you, but it essentially shows you the FDA rationale to launch Project Optimus, which is what I have been showing you on all those prior slides. So what are the potential benefits of improved dose selection at early stages? Of course, decreased toxicities to our patients. And decreased toxicities, hopefully, will result in decreased adverse events, decreased cost to the patients, decreased cost to the healthcare systems, improve go, no-go decisions. We'll see, but that's the hope. And faster drug approvals. On my summary slide, I will discuss with you some of the potential nuances of Project Optimus and some thoughts on what we need to keep in mind from several perspectives. So what are the challenges with dose selection strategy? On the left side, you have a classic dose escalation design, and you can see the FDA, it's looking at identifying a range of doses, not a recommended phase 2 dose, but a range of doses, dose level 2, 3, 4, that might be selected for further dose optimization in a randomized trial where patients are going to receive 2 different doses, and one of those is going to be selected to be tested in a registrational study compared to the standard of care. So that's the model the FDA is proposing now through Optimus. Just to summarize this part of the presentation, the recommended dose range is to be based not on a single piece of information, not just on toxicities, like what we were doing with cytotoxics, with studies designed to identify the maximum tolerated dose. But it's going to be a combination of the information about toxicity, responses, activity, pharmacodynamic data, and pharmacokinetic data. So there's different possible optimization scenarios. We can optimize during dose escalation, but we will need to also do an optimization in randomized phase 2. What are the limitations that we may face when we try to optimize during dose escalation? In a dose escalation study, you have patients with multiple histologies, pancreatic cancer, colorectal, lung, endometrial. So there might be additional variability that is introduced when you are trying to understand whether one dose leads to higher response compared to a different dose. Variability that is introduced not by the dose, by the different patient population. There's ways to control that. So you could definitely do a backfill cohort or an expansion cohort in a single tumor type to try to control that. In dose escalation studies, it's difficult to introduce stratification factors because we are limited in the number of stratification factors we can introduce by our sample size. If you are doing an expansion or expanding 2 dose levels to do different doses, and you're only including 15, 20 patients, it's hard to have a stratification factor, definitely hard to have more than one stratification factor. You can resolve that with a randomized phase 2 with a larger sample size that may allow you to use 2, 3 stratification factors. Why this is important? We know, for instance, for pancreatic cancer diagnosed de novo do worse than metastatic pancreatic cancer that relapse after a prior surgery. They are both metastatic, they are both pancreatic cancer, but the outcomes are different. Same for patients with metastatic pancreatic cancer that only have disease in the lungs. They do better than metastatic pancreatic cancers that have disease in the liver or the peritoneum. So there are multiple factors that will introduce variability that we need to keep in mind when doing this dose optimization. I'm going to use a couple of case studies to show you how dose optimization may work. This is a study with a different FGFR inhibitor, and bring you some examples from my wall, from the GI wall. Pemigatinib was approved by the FDA also for treatment of patients with cholangiocarcinoma with FGFR2 fusions. The phase 1 design dose escalation without reaching a maximum tolerated dose. And then part 2, you can see that they tested an intermittent dose level or a continuous dosing. In the next slide, what I'm showing you, it's some of the preclinical data suggested that hitting the target by causing a 50% inhibition of the target will translate into tumor growth inhibition. And with that hypothesis, they look at the dose levels on the right side, and when you look at that, you can see that those levels 9 and 13.5 on the lower bar on the x-axis led to 64% of target inhibition and 76. So they selected 13.5 milligrams because it led to a slight better target coverage. So what is the next step? So we're using here pharmacodynamic data to select doses, but how about activity? Look at activity. On this graph, you see that different doses, if you look at the bottom part of the graph, we go from 6 to 20 milligrams, and we can see that there's a peak in responses. We don't see more activity at 20 milligrams compared to 13.5. And now look at the table. What that table is telling you is that the higher doses did definitely impact quality. We see significantly more stomatitis and diarrhea with the dose of 20 milligrams compared to the 13.5. Based on that, the 13.5 milligrams dose was selected for further optimization. And this is the second case study with trastuzumab deruxtecan, an antibody drug conjugate that has been approved by the FDA for the treatment of several tumor types with HER2 3+ by immunohistochemistry. On this study, on the phase 1, again, a maximum tolerated dose was not identified. No DLTs were seen across doses ranging from 0.8 to 8 milligrams per kg. But there was one death, unfortunately, due to interstitial lung disease, an example of which you're seeing on the right upper part of your picture, with all those ground glass opacities that we see in patients with pneumonitis. So they did a dose optimization study in patients with HER2 3+ metastatic colorectal cancer, looking at 2 different dose levels. Higher dose level of 6.4 milligrams per kg and a lower dose level at 5.4 milligrams per kg, with a primary endpoint of overall response rate by blinded independent central review. And what did they find? Patients treated at lower dose fare better, despite this is a cytotoxic. The payload, as you know, antibody drug conjugates have an antibody against a target, in this case HER2, a linker, and a payload. The payload is chemotherapy. It's a cytotoxic. But this is targeted therapy thanks to that antibody. And despite this being a chemotherapy, they showed that the responses were higher at the lower dose and that the treatment-related adverse events were lower at the lower dose, 41 versus 50%. Similar to decreased toxicities, they also reported lower interstitial lung disease with the lower dose. So it's a no-brainer. The drug moved forward with the lower dose. So what are some of the key phase 1 design features? I'm not going to read you the slide because I'm going to go through each of these in the next slides. And someone told me I have only 10 minutes, so I think I'm good, I think, with time. So flexibility. Flexibility, it's a word I want you to keep in mind when you think about Optimus and phase 1 trials, because we have moved away from 3+3 design. 3+3 was a rigid design where we have a preset number of dose levels that we will explore during the phase 1 dose escalation, with only 3 or 6 patients. included in each of those cohorts, which will allow us to gain very little information because we could not get comprehensive understanding of the dose-response relation, the dose-exposure relation, and so on. Bayesian design is just one of the phase 1 designs you will see on your clinics, commonly coming with the new phase 1 protocols, and it's the paradigm of flexibility. So how does this Bayesian design work? It may incorporate an accelerated titration design, where you escalate dose rapidly using single patient cohorts, which is what this plot is trying to show. Dose level 1, no toxicities, increase the dose, sometimes even double the dose, go to dose level 2, no significant toxicity, or at least not grade 2 toxicities, go to dose level 3. And in dose level 3, you treat your first 3 patients, patient 3 to 5, and you get a DLT. What you have to do? You have to span, because your DLT rate, or what you're accepting as your DLT rate, has been passed, or you're hitting it. So you span to 3 additional patients. And now you have less than 30% DLT rate, assuming that was your DLT rate. So you can escalate to the next dose level. And in the next dose level 4, you enroll 3 patients and have 2 patients with a DLT, blue color. So obviously, you're above your maximum tolerated dose, and you have to de-escalate. Under a 3+3 design, you cannot go back to explore dose level. Under a Bayesian design, you de-escalate, enroll 3 other patients, show that that dose level is safe, and now you can escalate again to dose level 4 and continue to enroll patients at that dose level, and potentially continue to increase to the next dose level that perhaps it's more active. Flexibility. Also, when it comes to the design of the study and what cohorts we're including. Here on the right side, you have some preclinical data with one of the pan-RAS inhibitors that we have undergoing clinical trials at our institution. The preclinical data shows you that activity with this drug is not the same across different RAS mutations. What we see here is that with G12 mutations, the drug seems to work better. So when we think about trials, we no longer think just about tumor types. We need to think about the molecular profile, and we may need to explore in patients with different molecular aberrations. Perhaps patients with mutations in codon 12 respond better, as the preclinical data may have suggested, than patients with mutations in codon 13 or codon 61. We know from the study with BRAF inhibitors that BRAF don't work the same in colorectal and melanoma. So we may need to continue to explore different histologies. There might be sanctuary sites of disease. It's possible that for patients with brain mets, we may need to go higher on the dose so that we can see activity in brain lesions that we may not see with lower doses that may work for lung metastasis or liver metastasis. Patients with different organ dysfunction, like liver or renal. We may need to target a certain drug exposure, not just give patients X dose level, but see what is the exposure these patients are getting and implement therapeutic drug monitoring. And we need to take advantage of the backfilling cohorts to expand those dose levels where activity have been seen. Dose randomization. I gave you some hints before. On the left side, you have a dose escalation study where dose level 2 has been clear and activity has been seen. So for the next cohort of patients, we may randomize patients to go to the next dose level 3, or to stay on dose level 2, since we know it's active and patients are going to derive therapeutic benefit, to continue to gain a comprehensive understanding of the dose response and toxicities beyond what we can learn from just 3 patients from cohort. On the right side, we have a dose escalation design where we have identified dose level 2, dose level 5, and dose level 6 as those levels where we have seen activity. So we may do a dose ranging study where we randomize patients to any of these dose levels, again, to get a more comprehensive analysis. From Optimus, we need to come not with just a recommended phase dose, or most often not just with a recommended phase dose, but with a range of doses that we will explore further in studies that will allow us to optimize the dose before we get to a larger randomized trial. Some of the reasons I already mentioned in the prior slides. Intrapatient dose escalation. This is something we will not do in most 3+3, or we will not do in 3+3 designs, but we often see in Boeing and other designs. Why is this important? When we go to a phase 1 at a low dose level, it is unlikely our patients will derive therapeutic benefit. It is important we provide them with the option to be treated at higher doses once higher dose levels have been cleared, and once we know that the pharmacokinetic of a higher dose level is different to the pharmacokinetic of the lower dose level, and once we know that there's activity at higher dose levels. There might be other reasons to come with a range of doses. Certain tumor types may decrease absorption, may compromise entrance of the drug. Pancreatic cancer characterized by fibrotic reaction, especially at the primary tumor site. And we may need higher doses, or in theory, we may need higher doses of a drug to be able to access those sites. Certain molecular alterations, like I mentioned earlier, for GIST, we know depending on the mutation, we may need to use higher doses too. And of course, sanctuary sites of disease like brain and so on. Tumor biopsies. This is not something it's recommended routinely in the design of these studies, but it's critical that once we know what is the efficacious dose of the drug, we make an effort to at least obtain for a cohort of patients biopsy to understand what is the drug doing to the sites of disease, to get an understanding of how is that target downregulated, to understand that pharmacodynamic data that is going to help us make a decision when it comes to optimize the dose of the drug. When biopsies are not safe, we may need to look at surrogates of these tissue biopsies, liquid biopsies, looking at ctDNA, molecular imaging, and so on. Prandial conditions. Many of our studies don't allow dosing with food, and you know that when patients take a study drug and they are fasting, nausea is a problem, right? When they are dosing with food, we see less nausea. So we need to make an effort to allow these patients dosing with food, and then do the PK studies early to evaluate food effect during that dose escalation, and then modify prandial conditions if we need to. And of course, we need to broaden our eligibility criteria. We cannot do studies where patients do not represent the general patient population they are taking from, because then we cannot extrapolate the results of the research. This is a problem with pancreatic cancer, really many other tumor types, right? But particularly pancreatic cancer, some of the studies, very strict inclusion criteria. Patients need to have, for some studies, a certain albumin, right? And that leads to poor accrual of pancreatic cancer patients to clinical trial and delays understanding or developing new drugs. Ethical considerations. I touched a little bit on this, subtherapeutic dosing. That's why it's important that we allow this intrapatient dose escalation to give our patients the best option to see the benefit of the drug. Now, last slide. what are the challenges and what is in the future. So Optimus is designed to decrease, or hoping to decrease cost and to accelerate drug approvals. But obviously, there's going to be some stakeholders that are going to see increased costs and are going to see that perhaps approval, or at least getting to a phase 2 trial, takes longer. And some of them needs this to be able to get support for the next round of trials, small biotechs. So there needs to be some policies, there needs to be some implementation of things like, for instance, some tax cuts, some tax benefits to a company that decides to adhere to this Project Optimus, that thinks about the good for the community and what we are learning by having an optimized dose and avoiding the additional health care costs of taking care of all these toxicities. We need to keep in mind that some of these studies to optimize are clearly going to be underpowered, and that will lead to limitations in the information that we have and our ability to make decisions based on that information. But we also need to be realistic here. We're not going to come with a fixed dose in the absence of a randomized trial. What we're trying to get to understand from the dose escalation is just that range of doses. Interpatient variability. We can address this by implementing therapeutic drug monitoring, where we don't dose patients at a certain dose, but we are measuring the levels of the drug and see how we can customize treatment based on that address interpatient variability. And then, of course, there are different paradigms, like molecular targeted agents are not the same as antibody-drug conjugates. With antibody-drug conjugates, do we measure the antibody? Do we measure the payload? Immunotherapy, similar with bispecifics. When we go above the optimal dose, we may saturate the system, and we may not see the full potential of activity that we can see when we are delivering the optimal dose. And unrelated to this, I just wanted to bring you this front-runner project to your attention, with the hope of bringing some of these novel drugs to earlier stages of development, not treating patients, or not only treating patients who have undergone 7, 8 lines of therapy that may not allow us to see the full potential, the full benefit of that drug to our patient. And this is just my last slide, which I think it summarizes well what Project Optimus is all about. And with that, I just want to say thank you to our team. On the left side, you have many of our clinical research coordinators, research nurses, research data coordinators, specimen processors that make it possible for us every day to bring these treatment opportunities to our patient. And on the right side, 2 of my outstanding colleagues at Huntsman, Susan Sherry and Rachel Kingsford. Thanks for everything you do for us, but most importantly for our patients every day.
Thank you so much.